My name is Victor, and this is my presentation over my protein. My protein is DNA ligase, which is nicknamed it the litigating enzyme. So first, I'll go over like a little bit what proteins are. Proteins are very abundant in the cell. They compose about around 55% of its dry mass. And when I mean dry mass, is everything that's kind of not like made out of water or the aqueous or the cytosol or anything like that. I mean just pure mass. Proteins compose of 55% of that. Um, Proteins have very many diverse jobs, very different jobs. You can be a structural protein. You can be an enzyme. They can be signal receptors. They can be transport proteins. There's all sorts of jobs that proteins can have in the cells. Um, Proteins are made out of not DNA, amino acids. Um, these amino acids are, you have 20 amino acids found in proteins. Um, these amino acids all have a amino group, a carboxyl group, and a R group. This R group is what gives it this unique chemical abilities and structural abilities. So a protein will have an amino acid sequence that will conform into different shapes because of the way these amino acids react with each other. Um, because of this, proteins can be also called polypeptides because they're made out of amino acids, and the bonds between amino acids are called peptide bonds. Um, so structure enables function. Um, I talked about this a little bit before, but when amino acids come in a certain sequence, which they react to each other and they conform into a specific three-dimensional shape. This shape is what gives them this very diverse um, jobs. It can go anything from a signal protein to a structural protein to an enzyme, but it all depends on the shape of the protein, which comes from the amino acid sequence. One last thing is that the proteins have four levels of organization. They have the primary, which is the amino acid sequence; secondary, which is the alpha helices and beta sheets, whether anti-parallel, parallel, right-handed, left-handed. You have tertiary, which is the three-dimensional conformation of a singular polypeptide chain, and then you have quaternary, which not all proteins have, is when more than one polypeptide chains come together to form a larger complex. Um, so now my protein, which is DNA ligase, is an enzyme. And what enzymes do is they catalyze a certain reaction, uh, make it go faster, or make it go faster because enzymes can't alone produce a reaction. DNA ligase bonds at the 5' peptide of the DNA strand and the 3' hydroxyl group of the DNA strand, and they catalyze a phosphodiester bond between them. It kind of fixes the broken DNA. When DNA is broken by any means, DNA ligase is there to repair it by catalyzing a phosphodiester bond. DNA can split or break by very many different things. You can have oxygen-free radicals can attack DNA and break it. You can have the cell split itself and break its own DNA. You can also have gamma rays that affect the damage. They can damage the DNA, so you need DNA ligase for that. So here I have two DNA ligase proteins. So if you come a little here, there you can see like a little red dot. That red dot is the allosteric site of DNA ligase. Its cofactor molecule can be ATP or NAD, NAD plus, and then you also see the little pink little molecule here, which is it's actually a lysine amino acid. That's its active site. So going back to the cofactor molecules that DNA ligase can have, you have two main families. You can have a ATP dependent or NAD dependent. Depends on what its cofactor molecule does. What its cofactor molecule does is binds to the allosteric site and changes conformation so it's able to function. Eukaryotic cells are mostly ATP dependent. Some bacterial are also ATP dependent, but most bacterial cells are NAD plus dependent molecules. All cells have DNA ligase because DNA ligase is there to fix DNA. And all living organisms have DNA. They need DNA to produce other molecules and other structures. And they need a way to fix their DNA, and DNA ligase is there to kind of fix broken DNAs and join them together. The structure of DNA ligase, as I said before, proteins have 4 levels of organization. Here we're going to go over the 4 levels of organization for DNA ligase. The primary is the amino acid sequence. Its amino acid sequence is very important for the secondary and tertiary, which the amino acid sequence will give them that shape. A specific amino acid that we talked about before is the one on its active site, which is the lysine amino acid. Its secondary level of organization is alpha helices and beta sheets. As you can see here, there's mostly alpha helices, but it does have beta sheets, but not as many as alpha helices. Some DNA ligases can have 2 domains, while others can have up to 5 or 4 domains. These are 4 different types of DNA ligase, but they're still very similar in their conformations, even though they're for completely different organisms. Its tertiary is this their conformation is they all have a C-shaped conformation. As you can see here is a C-shape here, C-shape, and they even though they're all different DNA ligase, they have a similar conformation because they all do basically the same job, which is ligate the DNA. It's just these are different molecules. We require slightly different ones, but they all basically have the same tertiary conformation. Quaternary conformation, DNA ligase has no quaternary level of organization since it's only made up of one peptide, polypeptide. So there is only one, so there is no quaternary. As I said before, DNA ligases structure remains similar through the many different species. That is because they have the same relative function. Genetic information of DNA ligase. DNA ligase genetic information has been studied so much because of its many research applications that it can have. We can research DNA through DNA ligase. It's very versatile when studying DNA. People can use it to diagnose viruses and all sorts of things. This is why DNA ligase has been cloned and its nucleotide sequence has been discovered. I would like to, because there's many different ones, many different DNA ligase, I want to focus on TIF DNA ligase, which is has been studied a lot. I just want to give you more precise numbers. So TIF DNA ligase has 667 amino acids. Its molecular mass is 75,936 Daltons, and And even though there's many different types of DNA ligase, TIF DNA ligase is very similar to other ligase. For example, TTH DNA ligase. Their genetic sequence is about 86% similar, and even an E. coli DNA ligase is 46% similar. So DNA ligases they are all relatively similar again because they do about the same thing. There are no relevant illnesses related to mutations in DNA ligases. This is because the body can produce so many, and there aren't very many, and it's also very dangerous if you have a coding of a bad DNA ligase because that means you can't fix your DNA, so you're probably gonna die at a very early stage. But DNA ligase can be used to diagnose other illnesses. For example, you have HBV, which is a virus that changes a cell's genetic material. So what scientists are able to do is, with DNA ligase, they're able to magnify that DNA. They're able to see how it functions through DNA ligase, and they're able to identify mutations early to diagnose people with HBV, which is a very fast moving virus and has very serious implications, and it can lead to death. And yes, that would be it. Thank you for listening.